The overall goal of the following experiment is to convert cells derived from human brain pericytes into induced neurons. This is achieved by isolating and culturing pericyte-derived cells from surgical brain specimens. Next, following expansion in vitro, cells are immunolabeled for parasitic markers, which allows for the characterization and eventual further enrichment of the cells. Then, Pericyte-derived cells are transduced with retroviral vectors encoding neuronal fate determinants in order to directly convert them into induced neurons. Results are obtained that show induced neurons obtained from lineage-reprogrammed pericyte-derived cells based on immunostaining for the neuronal marker beta-3 tubulin.
The main advantage of this approach over existing methods like reprogramming of fibroblasts or differentiation of embryonic stem cells into neurons is that we target cells endogenous to the brain, which may ultimately pave the way for direct lineage conversion within the brain without having to transplant the cells.
The following protocol was developed in accordance with the approval by the Ethical Committee of the Medical Faculty of the LMU Munich and written informed consent from all patients. Beginning with a fresh human brain biopsy, keep on ice in HBSS and heaps. Dissociate the tissue into single cells by transferring it into a 65 mm Petri dish and using 2 sterile single-use scalpels, mince it into small pieces. Transfer the tissue to a 15 ml conical tube and add